Professor, please explain to me what led you to oppose Dawkins' selfish gene concept.
Well, Curt, I think it's not so much to oppose. I think one has to dig into the history of biology in the 20th century to understand that perhaps the selfish gene view of Dawkins is incomplete. And I think what led me, as you say, to oppose, not so much to oppose, but to appreciate or to identify some weaknesses in that theory is my background in developmental biology. I am interested in how animals are built, and they are built from the zygote. And you might recall that a very important tenet in the views of Dawkins about the selfish genes is that organisms are instruments that genes build in order to travel through time in a way. They are just structures that are a direct consequence of the genes so that the genes can compete with each other to travel in time. But I think that the problem with that view is that it ignores completely developmental biology. I don't think there is anything wrong with the selfish gene view of Dawkins other than it's very limited in what it explains of biology. The selfish gene view of biology, which is the gene side view of biology, it's a consequence of the developments in the 20th century to explain biology in terms of genes. If you look at the history of biology, it starts the 20th century with the discovery of the gene, which becomes a very powerful concept that one can use to explain a great deal of things, okay? It's interesting that for 50 years, people don't know anything about the material basis of the gene. And the gene comes into being as a contradiction, actually, to Darwin's ideas of evolution, because Darwin describes an evolution that is continuous, in which the phenotypes there is continuous variation and selection acting on that continuous variation. It's what starts creating the shapes and the structures that we see around the world. He didn't have an explanation for the material basis of this continuous variation. When the genes come, they provide something of an explanation, but they create a problem. And that problem is that they are discrete units. And the question is, how can you bridge the gap between a set of discrete units and continuous variation? And that is the great triumph of population genetics and what is called the modern synthesis, which some people today are trying to challenge and say that it needs to be overturned. But it creates a connection between these discrete entities, which are genes, and the continuous variation which occupies the world.
Can you explain the difference between cell biology, population genetics, and then developmental biology?
Yes, I'll try.
Are these different views? You mentioned there's a gene-centric view, and I assume that's what you're opposing, or that's what you're disputing as incomplete.
That is, I'm disputing that is the whole explanation for biology. We live in a time where wherever you look, and I'm sure you will agree with me, everybody, you open the papers and everything is about our genes. It's about our DNA. There is personalized medicine, which measures the genes and will give you medicines according to your genes. People talk about the DNA of a company. Mm-hmm. People talk about it is in your genes. I mean, it's absolutely pervasive, okay? And actually, there is a whole bunch of things that are not in the genes. I mean, and I can tell you, someone will come to that in a minute. So the gene view of the world is that this molecule that is called DNA, where there are these stretches, which are called genes, which code for proteins, is all there is. That if you know the genes of a person, you know that person, okay? That is really what they are telling you. 23andMe, for example, will sequence your DNA and will tell you a lot of things about yourself that you might believe or might not believe. So the view of biology from the gene is that if you know the genome of a person, you know everything about that person. In fact, it's been said, now there are some people that are backtracking, that if the genome contains a blueprint of the organism, that all you have to do, there are instructions there to build an organism. It's nothing of the like, okay? So genetics, genes, they just look at this sequence of DNA. There is a blueprint in DNA, is to make another molecule of DNA. That's about it. That's because there is a template, there is the double strand, and one strand can code for the other, and that's about the only blueprint. Cell biology, it's an old discipline, and what it tries to understand is the structure and function of cells. Cells are very complex structures, particularly what we call eukaryotic cells. We are made of cells that are eukaryotic cells. Prokaryotic cells are bacterial cells, which are much simpler. They are not so complicated. Cells have an enormous repertoire of behaviors and an enormous complexity. Cells are really an emergent structure. They emerge their behaviors. They really, you cannot predict from their components. In fact, one of the points that Dawkins always makes, and the people that suggest Dawkins, is that DNA is a great replicator. It replicates itself and passes from one organism to another. Is the only thing that can replicate. But actually, there are many structures in the cell that cannot be made out of genes only. They have to be replicated in order to be themselves. Membranes, for example, you cannot create. If you put together all the genes that are supposed to be involved in making membranes, they will not make membranes. A cell needs another cell in order to recreate. So cell biology is about this emergent structure that we call a cell, which has a number of properties that are not in the genes. It can interact with other cells, it can divide, it can grow. It uses the genes as tools to do all this. But it's not, a lot of its properties are not encoded in the genes. They are emergent from the interactions of these structures that appear from the genes that are the proteins. And to finish, developmental biology, what it does is studies how cells interact with genes in order to build organisms.
I see.
Those would be the three elements. Whereas in a very gene-centric view of the world, genes have a blueprint. In your DNA, there is a blueprint for the organism. The way I would think it is that the genes are coding for a number of tools and materials that the cells actually dip into in order to build organisms. They are constantly dipping in those cells. And I think the understanding of these emergent properties and how cells interact with each other is something that is now coming into the fore, which can explain many things that a gene-centric view of the world it can't. I think if you want to say, when we think about an organism, if the organism is driven by internal forces or it requires something else that comes from the outside, I think obviously it needs something from the outside. As we were talking, we are dissipative structures. So this is something that needs to be addressed. As we can do measurements, people are able to start, they're starting, but they're able to do thermodynamics of certain developmental events and doing measurements.
There's a concept called genetic determinism. So would you say that you disagree with that?
Completely. I think this is one of the errors. If you talk to geneticists, nobody will own that concept. They will say that nobody, but actually, when you look at their actions, and I think actions is what matters.
Right.
This is a very extended view, and I'll give you some examples.
Please.
And I think this is where I think we need to think at this moment. Genetic determinism is the notion that we are determined by our genes. In a way, if you wish, is an extreme consequence of the selfish gene hypothesis. We are nothing but our genes. I am not refuting. I want to stress that there is anything wrong, that the genes have something to do with us. I'm just saying that they don't explain everything, and that we have to extend this and to think a little bit deeper. The idea of genetic determinism leads to eugenics in the early part of the 20th century, which is the fact that having decided that we are our genes, we can decide who is the perfect human being and get rid of those whose genes, defined in a very abstract manner in the connection between genotype and phenotype, we can get rid of those people. And that's what the practice of eugenics was, particularly in the United States, where it reached the point that even certain immigrant groups were said to have bad genetics, and they were judged to be able to enter or not in the United States, depending on this. So genetic determinism, the first incarnation in the beginning of the 20th century, leads to all the horrors of eugenics, which now everybody will abhor and they don't want to abstain. Then we enter into a phase where we now agree that we cannot characterize an individual by one gene or by one or a small group of genes. But with the emergence of the Human Genome Project, with all this ability of getting genes in a very cheap way, having accepted that there is not one-to-one correspondence between a gene, and I'd like to qualify what do you mean by a gene now. A gene is a stretch of DNA that has many different forms, that is called alleles. People sometimes talk about having one gene we all have the same genes. The variance of those genes is what creates the differences. So somebody that has sickle cell anemia is not because they have the gene for sickle cell anemia, it's because the gene that codes for myoglobin or hemoglobin is defective. So this idea sometimes that people have a gene for a disease is a misunderstanding. We all have the same genes. It is the versions that we have of those genes that can be faulty or can be different, let's call it like that. You read in the press, this person has the gene for cancer or this person has the gene for diabetes. We all have the same genes. It's the variance, it's the colors of those genes that are different between us. And some of those colors can cause disease on some occasions, okay? The number of diseases that are monogenic, that can be linked to a gene, to one gene, there are many, but they're not very representative. Given the number of diseases that we have, there are not that many. So what people have done is create this notion that is called the polygenic risk score. Polygenic risk scores.
Risk score, got it. Okay.
It's a statistical quantity that tries to identify the genetic contribution to a particular trait in the population. Okay? I'm not going to go into the technical details, but this is now very important because people are sequencing genomes, and on the basis of your sequence, for example, recently they got very excited because they found the number of genes that they have to measure to determine height. Okay? And they call that they have 10,000 genes. To me, that doesn't make any sense. They are translating a genetic determinism based on one gene to say now we can make a statistical measure over thousands of alleles and now give you a probability of you being 170 or having diabetes or having... And I think this is very serious because usually what is called the heritability, the contribution of the genetics to this character is very, very low. But this is now a new form of genetic determinism in my view, okay? Uh-huh. It's a form of now rephrasing. Instead of one gene, we are going to find a statistical measure that gives us a number to say how much of a genetic contribution is for you, and we can tell whether you're going to be sick or not.
That sounds like a natural extension of statistical mechanics applied to the genes. Why do you not like that?
In a way it is, but not to phenotypes, is to genetics. And these polygenic risk scores, now they are all the rave. In fact, last year, there was a very famous book in the States called The Genetic Lottery. There is these people called behavioral geneticists, which claim that on the basis of these measurements, by sequencing the DNA of a child, they can determine what their school attainment is going to be. Uh-huh. And somebody in a book called The Genetic Lottery proposed to reorganize the school system in the United States based on polygenic risk scores that they can assign to possible attainment. To me, this is genetic determinism. This is a second version of genetic determinism, and one that is very, very dangerous. In the UK now they are sequencing the DNA of about hundreds of thousands of children in order to try to assess the probability that they are going to have diseases on the basis of these thousands of alleles or colors or shades of gray, and then you can intervene or not. I think that there is a danger in this because we don't understand. As I said, this is in a way an extreme form of the Dawkins that the individual, the organism, is a linear consequence of the genetic makeup. And this is very dangerous because between the gene and the organism, there is the cell. There is a great integrator, there is a great worker, there is a great architect. I wrote this book, The Master Builder, which is all about this dichotomy and about highlighting the things that genes cannot explain. Nowhere is written that we have two arms or we have five fingers. There is nothing in the genome that says that. There is nothing in the genome that positions Arise. As I tell in the book, our fingerprints, we have 10 different fingerprints. There is the same DNA. That's why you can only open your phone with one finger. The one of the other hand will not do, because your fingerprints on this hand are unique. They all have the same genes.